welcome to the Iowa Grazing Academy. These are the recordings from the 5 online sessions that were held in July of 2026. If you have any questions, please contact Shelby Gruss at the email address listed on the slide. Thank you and enjoy these sessions. Hello and welcome to
I will address nitrate toxicity when utilizing annual forages, and Shelby will get into the discussion of prussic acid. So I know Dr. Radke talked about nitrate toxicity a bit with fertilizer and water cases, and I would say that's by and large the most common situation we see. And I'll get into why, what we know of nitrate toxicity, how it does and doesn't necessarily apply as straightforward in these grazing situations when we're utilizing annual forage. So first and foremost, understanding how nitrate toxicity works is really important in understanding and evaluating the risk you might have in various situations. So nitrate, it's in all plant material to some degree, right? The plants, the roots take up nitrate to utilize, and they utilize that to make protein. So it'll accumulate in the lower portion of the plant, and then when the plant is able to photosynthesize, it'll convert those nitrates into protein and disperse it throughout the plant. But when cattle or other ruminants consume plant material, nitrate will be entering the rumen. Nitrate is a water-soluble, really readily absorbed material, as well as nitrite. So the nitrate enters the rumen. It is reduced to nitrite by bacteria. The bacteria consume and convert that to nitrite, and then it is further reduced into ammonia, which can be utilized as a protein or as a nitrogen source for bacteria, which will eventually turn over into microbial crude protein available for that animal to utilize as a protein. So when we have an issue with nitrate toxicity, what happens is this nitrite to ammonia step is considered a rate limiting step, meaning that reduction occurs at a slower rate than the nitrate to nitrite step. And so when that happens, this nitrite material or compound will accumulate, and it is also water soluble and readily absorbed across the rumen. And as it accumulates and absorbed across the rumen wall, it will bind to hemoglobin and convert that to methemoglobin, which cannot carry oxygen. So in nitrate toxicity, all of the clinical signs and essentially what causes issues or kills the animal is that suffocation. So even though they're breathing, the blood can't carry oxygen. And so really when it gets to nitrate toxicity, we're really talking about nitrite being the issue and being converted at a slower rate. So kind of history, I guess, of nitrate toxicity will help in our discussion throughout the next half hour, but it was first recorded in 1895. It's a pretty well-known toxicity risk and has been around or was observed a long time ago. The studies to start determining like the toxic levels of nitrate really didn't begin until about 1940 in Wyoming. And the first LD50, which just stands for lethal dose 50, suggested was 1.5 potassium nitrate. And for the sake of discussions today, I have everything converted to 2100 milligrams of nitrogen per kilogram of dry matter, or that's also equivalent to parts per million of nitrate nitrogen. So based off of Bradley's work, he suggested a lethal dose rate or toxicity guideline to be set at around 2100 parts per million of nitrate nitrogen. When they did this research, all of the dosages of nitrate were administered via a stomach tube with potassium nitrate, basically in a bolus type form. And so if you remember on how previous with the rumen, how nitrate toxicity works, nitrate is really readily available already and water soluble. And so that is a dose that was immediately available in the rumen and quickly overwhelming the rumen, right? When we got to 1960s, Davison and others kind of started looking into any reproductive effects that nitrate toxicity or nitrate levels might have. And this is really kind of the first time frame the reproduction side was considered. And if you look at today's guidelines, a lot of extension publications or articles will talk about um, not feeding to pregnant animals because of an abortion risk. And in that study, he dosed it at two different levels by spraying the or top dressing hay with ni potassium nitrate salt dissolved in water and sprayed onto the hay that was fed at known levels. And so dose close to the 2100 and then 3300 parts per million of nitrate nitrogen. And in that, he didn't see any deaths. He only saw one abortion at at the higher dose. And so that was the first time that was really kind of recorded. I'm not going to talk about this much, but I could definitely get into we really don't see abortions in nitrate toxicity cases bef where the cattle aren't already really close to acute toxicosis and death. And so if they're starting to experience an abortion, they're really close to dying from nitrate toxicity at that point. And then we haven't readdressed the research very much to try to reestablish lethal doses. Crawford did attempt to get it a little more refined in the 1960s, and he did at least compare a drenched dose of nitrate versus top dressed on the hay. And he did notice that when it was top dressed instead of directly administered into the rumen, that the cattle tolerated about a 3 times higher dose. And so even though recommendations didn't necessarily change, he suggested that consumption rate is a big factor in nitrate toxicity. So I wanted to at least touch on the history because it gives us a lot of background on where we're at today. Because yep. When we look at nitrate toxicity today, I just pulled, this is from Iowa State's nitrate toxicity publication that's most recent. But it is pretty well dated at this point. But by and large, the majority of extension publications actually are more conservative than that first toxicity guideline that was suggested by Bradley and others in the 40s. And most put the generally safe category as up to 1500 parts per million. There's some differences across universities or extension publications. Some of them will discuss management factors and considerations that can change that, but really all of them kind of have that 2100 is like the maximum that they ever consider as safe. And essentially today, more than anything, being aware and understanding nitrate toxicity can help, especially in grazing annual forage scenarios. We can actually, I would argue those guidelines need to be updated, but also hard to do. So there's Lee and others did a nice review on nitrate toxicity that I liked, that he broke down into 4 different categories that really drive the risk of nitrate toxicity. The first, being the most obvious, is nitrate levels in the diet. Second being the nitrate consumption rate. Third being nitrate and nitrite reduction rate. Remember, nitrite is that compound that really accumulates and causes the actual toxicity. And then finally, rumen passage rate and how fast that's all processed through the rumen. So why are today we're talking specifically nitrate toxicity in annual forages, and why is that kind of a primary concern? In reality, annual forages are known as nitrate accumulators, especially brassicas. A lot of times, and when we talk about benefits of cover crops, that's actually a really good benefit that some of these species can do, is to accumulate and tie up those nutrients a bit better. But how often are annual forages, do they test as toxic? So part of my work at UNL with Dr. Dranowski, my maiden name was Lenz there in the citation, but we got a bunch of samples from Ward Laboratories in Nebraska. That was a commercial laboratory over a 5 or so year period. And I took all of those, narrowed them down to what I could identify as an annual forage in these 5 categories. So brassica, cover crop mix, small grains, millet, or sorghum sudan. And then I just looked at what percent of those samples fall in this safe range of under 1400 parts per million nitrate nitrogen, marginal, cautious, starting at like that 2100 suggested dose, and then that all all publications would recommend not utilizing a above 5,000 parts per million nitrate feed. And so when we looked at that, brassicas, I commented that they're known accumulators. 48% of the samples would have been caution or toxic category. But even those that showed the least likely to accumulate nitrates, or to not accumulate nitrates, least likely to test in those toxic levels, still had 20% of the samples that high. But we do have cases of nitrate toxicity in annual forages, but it's not near this frequent. And so kind of why, and that's what we'll get into. The next slide, I think, is a good, also while I was at Nebraska, kind of retrospectively looked at a bunch of annual forage trials and took those forage samples that they had and ran them to test the nitrate content. And even though those trials were done, we never saw any visual nitrate toxicity signs, never had reasons to be concerned that there might be a risk based off of growing conditions and things. But if you look, they frequently tested for high nitrates. I'll kind of explain or a caveat on why they kind of tested, because consistently sampled at ground level. So that's kind of the worst case scenario. But these were consistently testing high, and we really didn't see any health effects. And I just an interesting note. So there is the risk, but how concerned should we be? Is kind of that question. Again, it gets back to those 4 factors that I'll talk about how it'll mitigate that risk a little bit or just drive how much risk there is. So the first being nitrate in the diet, we've talked a little bit about that, but the plant part is a huge portion. So the majority of nitrates that accumulate in the plant will be in that bottom third of the stem. And that's why I said on that caveat to those nitrate results, those samples are taken at ground level and are definitely the worst case scenarios of what that animal could consume. Also consider for what influences nitrate concentration in the diet. Plant species we talked about a bit being annual forages, but fertilizer treatment are available and in the field. If it's in a cropping system like a seed corn or corn silage, there might be some residual N left that those annual forages will scavenge really well. Forage maturity makes a huge difference. So the younger the plant, the higher the nitrate concentration. And so delaying utilizing that forage until it's farther along or more mature is one effective strategy. Weather also makes a difference. And I like explaining this as it's really those events that cause plant stress or drought, cloud cover, hail, frost, all of them kind of come into cause nitrates to accumulate similarly in the fact that that plant is constantly taking up nitrogen from the soil. And so when there's any type of weather condition that hampers that plant's ability, to photosynthesize, that's when we have accumulation occur. So if drought, those nitrates are accumulating, but really our biggest risk would be right after a drought relieving rain when it's finally starting to move through the plant, but hasn't had time to photosynthesize and process. Other dietary sources to pay attention to: water. I think Dr. Radke kind of hit on that, but it's over 10 times more toxic when it's in the water than when it's in the feed. And that is directly correlated to the fact that it is water soluble and readily absorbed across the rumen. So even like a feed, when they eat the feed and it gets into the rumen saying it's readily available, but the bacteria have to kind of break down that plant cell wall in order to have access to that at all. When it's in the water, it's available right away and really overwhelms the rumen, absorbed directly. And then also other feed stuff. So if you're feeding other things or supplementing while they're on these annual forages, does that additional feed dilute or add to the nitrate load in the rumen? Consumption and availability kind of being, these aren't exactly the 4 points I said earlier, but I'm hitting on them, I guess. But slower consumption will help keep that rumen from being overwhelmed. So I think we've talked a little bit about that already. A really interesting study I thought that was a nice demonstration on grazing forages, why animals might be able to tolerate higher risk. But the Gering in the 1970s took chopped up hay with a known nitrate concentration and chopped up fresh grass with a known nitrate concentration. He put those both in water and let it sit for 20 minutes. And just based off of the fresh or dry plant cell being more permeable, so like a dry plant cell is already kind of cracked and more easily accessed and broken down. Um, in that 20-minute period in hay, 80% of the nitrate was released into the water, and then a fresh forage only 30%. So there's some of that coming into play as well. And also, plant part is huge, top-down grazing. Leaves have less nitrate than that stem, and then a lot less than that lower part of the stem. So as you allow animals to be selective and don't have high stocking densities forcing them to eat that bottom portion of the plant, that'll really help mitigate any risk of nitrate toxicity you might have. And last couple points on that, nitrate and nitrite reduction and also passage rate through the rumen. So they're kind of breaking that into 2 points I want to talk about. Adaptation to higher nitrate diets is definitely possible. Not really an example of how to do it or how much you should try to do it, but a really good demonstration project was done in the 80s where they increased the nitrate level in some sheep diets, just top dressed hay in a feed bunk scenario every 2 weeks until they got it to be 5 to 10 times the recommended toxicity threshold, depending on whose recommendations you were going off of. And the sheep didn't have any health issues in that adaptation period. But what they did towards the end of that is they took rumen fluid from those sheep, and those adapted animals, they reduced nitrate 3 times faster and nitrite 5 times faster than the unadapted animal rumen fluid. And then finally, diet quality has a really big role as well. Energy supplementation to support the bacteria will help them convert that nitrate and nitrite more effectively. And also another like interesting side note, that lower or energy supplementation will eventually decrease your pH some. I'm not saying to manage to a 5.6 pH by any means, but a 5.6 pH is ideal for nitrite reduction, which is that rate limiting step, versus a higher, like I think it's 6.2 pH is what's ideal for nitrate reduction. And so energy supplementation kind of helps in both of those. And a comment I want to make because of that, we're talking annual forages and grazing annual forages. These are really, really high quality feeds. Brassicas, even though those were our example of really likely to be toxic plants, the tops of brassicas are actually really comparable to dry rolled corn in their energy content, which I don't think a lot of people necessarily think about on how high quality these feeds are. And to kind of wrap up, like overall, for grazing annual forages, the current recommendations, my opinion, are definitely too conservative. But as a producer, I mean, you don't want to take on any risk of losing cattle, right? It's really important to go through and acknowledge the risk. I really encourage testing a forage just to get a baseline and understand what you're working with. But you really need to understand what the water risk is. And then we've talked about most of these mitigation factors on reducing risk, but letting them be selective, turning them out on a full stomach so they don't eat really fast and indulge, and things like that. So I know I used up most of my time, and Shelby's got to get to talking about prussic acid. So drop questions in the comments, and yeah.
All right. Oh, all right, real quick, we're just going to talk about prussic acid real fast, and why we chose these two is probably get the most calls on these two is prussic acid and nitrates, especially as we start introducing summer annuals and cover crops into the mix. And so prussic acid is specific to sorghum species, so forage sorghum, sorghum Sudan grass, and Sudan grass. And when I talk about it, I generally introduce the topic of Durin, and then why I care about Durin is Durin accumulation is what contributes to hydrogen cyanide poisoning of livestock. So Durin is the precursor to prussic acid release. And so how this works is it's kind of similar to nitrates in the sense that the animal kind suffocates in a way. And so as an animal eats a sorghum forage of a type, it contains durin, and as they chew, that breaks down cell walls that introduce durin to durinase, which can release hydrogen cyanide, or it can be released in the rumen with microbes that are in the rumen. And as it's released, it enters the bloodstream and it's carried throughout the body to every tissue. And what it does is it basically stops cellular respiration. So the cells cannot use the oxygen. And so the blood's able to carry it, and that's why we get this bright cherry red blood with hydrogen cyanide toxicity, because the blood can carry it, but the cells, when it gets to those cells, cannot use it. And so it can happen really rapidly. And so why do these plants have Durin and this risk involved in it? And so this work came out of Australia, but they did this really intensive sampling looking at where Durin is accumulating throughout the plant and then also the enzymes that break down Durin into hydrogen cyanide to kind of get an idea of why it's used and why it's there in the plant. And the big one is Durin is high in young leaves and young vulnerable tissues. So that defense mechanism where mostly where we think about why Durin and hydrogen cyanide is produced in these plants. And so we see that in high in young tissues and vulnerable tissues, typically younger tissue, plant tissue. And so we have some recommendations. Don't graze it when the plant's small. And we can see it that it's typically higher in the leaves in comparison to the stems. There's some other theories why Durin is there. And the last one that I really just want to point to is recycling. We think it has something to do with nitrogen mobilization in the plant. And so we can start to see Durin accumulation when we apply high levels of nitrogen. And so when is it accumulating in the plant? And the big one, like I mentioned just previously, was that young tissue. And this study that was done, they were looking at Durin basically concentration and then the total amount of Durin production per kilogram or Durin accumulation per hectare. So this is on a kilogram basis of Durin per hectare. And what you see is as that plant gets bigger and taller, they had three sorghum and Sudan grass varieties that you can see the concentrations of Durin in those plants decreasing over time as those plants grow. Typically, that's what we think of. It typically is a dilution thing as the plant grows. What I think is interesting is that over the whole hectare, we still have similar levels. It does decline some throughout time, but it's a little bit more similar, but it's a dilution issue. Typically, most of our plants do have some Durin in them, but we're not at toxic levels. And so it's as that plant grows, we see that that accumulation of Durin, that high concentration decline, and those plants become safer to graze. The next one is that stress tissue. Frost is a big one here in the Midwest, and we're going to dive a little bit more into that data in just a second. But another one that I wanted to point out because it seems like we're entering more and more drought situations is that there was a study done in 2020. They were able to see that in an extreme drought situation, and these were very droughted plants, and with the addition of high levels of nitrogen, they really did see an increase in Durin accumulation in those plants. And so that plants again is going back to protect itself. So unfortunately, some parts of the United States right now are facing high levels of drought, but we need to be careful in considering what to do with those plants in those cases. And then we also have cultivar specific. There is a wide range of variation in how much a plant will accumulate. And I like to think about even species wise. Typically, species wise, I think of like Milo, that sorghum grain, typically has the highest risk of grazing. Where if you go down all the way down to Sudan grass has the lowest risk Durin accumulation. And so when we're looking at frost, this study was done in 2020. We had it in 4 locations. This was in Indiana. These 2 were in Kansas. And what we saw in Indiana is that following a frost day 0, this is the day prior to the frost, Following 3 days later, we saw this increase in during accumulation. It basically doubled due to that frost. And this actually photo in the back is from that trial. And then 7 days later, it started to go back to normal. And so if we go back to that, our typical recommendation is we have a frost, we wait 7 days, and that should be safe. But what we saw in Colby, Kansas, and Garden City really points that this is a rolling suggestion because we saw from day 0 to day 3 to day 7 a continual increase in during accumulation. And at these two locations, we saw a continued frost occurring throughout that week. Where in West Lafayette, we saw that there was one frost and we went back to pretty high temperatures so that that plant stabilized again and went and decreased in during accumulation. Where at Garden City and Colby, they continued to have frost conditions. And so what can we do and when can we graze sorghum just to make sure that we're doing this safely? And so avoid grazing plants that are small. So that young plants, we typically think of 18 to 24 inches. Don't graze something that's less than 18 inches. Extremely drought stressed plants can be problematic. Frost stressed plants is our big one here. And again, that is a rolling suggestion. So we wait a week, and if we have another frost occur, that week restarts. We can also use cultivars known to be low in durin accumulation. There is a durin free out there now that does not produce durin at all, so we don't have that prussic acid concern. And then finally, we can test as well. Now, if we are concerned, what can we change our management strategy to fit this? And the answer is yes, but we also need to consider hay, because hay, we have found that it actually does not reduce it. So if we do want to harvest this differently instead of grazing, silage is our best bet. It can decrease durin and prussic acid potential by 50 to 80%. Grazing, we do not have any reduction. So unlike nitrates, where nitrates is in the lower stem, prussic acid and durin accumulates typically in the leaves. And so generally they select for leaves, so it can be more dangerous or risky. Hay, again, we did not see any reductions. This is some more recent work that occurred. So haying your sorghum Sudan grass to reduce prussic acid is not a good option anymore. And so finally, just testing it to make sure it's safe. You can send it into a lab to make sure that it is safe. Most labs do a good job and add this what's called an external beta glucosidase. This is important to ensure that we're evolving as much HCN from the plants as possible to be able to get the value. This is kind of what it looks like in interpretation wise. As we start to get to dangerous levels, we're looking at over 500 parts per million of hydrogen cyanide toxicity. And then if we're over 750 to 1,000, it's very highly toxic. There is an at-home test from Ward Laboratories. If you were to do this, just making sure you collect fresh tissue, you mash it up really well. They have a great video to watch to be able to do this at home. This is a good option to basically make sure, yes, safe, be careful, or please don't graze. It's just a semi-quantitative analysis. It just kind of gives you a range of potential. But with that, I know it's after 1:30, so if there are any questions, I'm happy to stay on to answer anything. So please feel free to unmute or send it in the chat.